देखो ईरान कहाँ और मुजम्मिल कहाँ।
हमारे बनाने लिखी है।
हाँ।
अगर उस
मुझे
हम
इसको
लोग
करना भी क्या है।
अगर उससे हम लोग
आपने तो खुद ही देख लिया ना, हर एक बच्चे के मुंह पे रुचिका चढ़ी हुई है।
अभी क्या पहले
वालों के
सारे
क्या, अभी वालों के क्या।
हाँ नहीं मैं करती हूँ, आज इसका हम कर देंगे। ये क्या है?
कहाँ हो सोनिया? ठीक है
चलो मैं अभी क्लास ले लूँ।
हाँ हाँ
मेरी है क्लास, फिर मैं आपको फोन पे मिलती हूँ।
हाँ ठीक है, फिर आप आ जाओगे
हाँ
कल आ रहे हो?
आ जाना, क्योंकि अब ना लड़कियों का भी इशू हो
बहुत
रहा है।
ज़्यादा। नहीं आज या कल। मैं कल आप ना उधर रख देना, बिस्तरा बिछा देना, अगर जब भी टाइम मिला मैं आ जाना। मैंने कल 10-11 बजे पहुँच जाना है, बैग ही उठाना मैंने, बस मैं बैग उठा के मैं पहुँचूंगी। हाँ और बाकी हम बात फोन पे करते हैं। तो मैं ना क्लास ले लूँ, पर मैं इसका करूँगी यार, हम बैठ के करेंगे एक दिन, बैठ के मेरे दिमाग में है, बैठ के करते हैं इस चीज़ को। हाँ
इनको ये लग रहा है सब रिपोर्टिंग करनी ही नहीं है, करनी है एक चीज।
हाँ बात तो सर आई नहीं अभी तक। चलो कोई बात नहीं, फिर मैं सर से खुद बात कर लूँगी। अभी ना मेरी क्लास है बच्चे, तो मैं सर के पास जाके आओ, फिर मुझे बताओ, या उस दूसरे नवजोत सर के भी पास जाओ, मुझे बताओ फिर। Hello. So very good afternoon to all of you. My voice is audible. Please let me know. If my voice is audible to all of you, please let me know. If my voice is audible to you. Okay. Thank you all. So how are you all? Please let me know. We have Sadhan, we have Merlin, Sandra, it's Radha Lakshmi, Rajneesh. So very good afternoon to all of you, Bhavna. And yes, tomorrow. Tomorrow I will take your session at 4 or 4:15 p.m. Because some students have some exams or something like that, so we will take it on time. So I welcome you all. I hope you all are fine and doing great in your life. Yes, we have Rajneesh, Shaina. I think Shaina Gautam has maybe practicals or something up to 4 p.m. I think so. Some of you have. Okay. Sorry, Shaina. No. Yes, it's Radha and Apurva. Yes, it's Radha and Apurva. So for Radha and Apurva, we will try to take our classes. Tomorrow we will take at 4 p.m. or 4:14 p.m. because tomorrow I am in my home, so I will be able to take the class. So yes, do you have any query or anything to say? Please let me know. Otherwise, we will start the class. Yes, Tejaswini, good afternoon. How are you? So just let me know if you have any query or something. Otherwise, we will start the class. We will just allow some more students to join. Then we will start the class. Yes, good afternoon, Samdeep. How are you? Ayushi, good afternoon. So, do you have any query? Have you selected your topic for review paper? Please let me know. Have you selected the topic for review paper? Is everything is clear to you? I'm really sorry because I'm not able to read your telegram messages. I'm really sorry regarding it. But please let me know. Everything is fine. Are you able to, you know, are you able to get everything regarding the review paper? Please let me know. Yes, I already give the topic in your Telegram group. I already provided to you. You can select any topic of your point of interest. Please let me know. Okay, Madhuta. Madhuta, it's very difficult for me to explain again. But yes, I already give some topics for all of you to write a review paper. Even you can select any topic of your own point of interest. You can select any own topic and you can continue to write the abstract. Yes, yes, Pooja, definitely I will reply. Yeah, Kalpesh, today I will specifically focus on to tell all of you how to write the review paper. I will let all of you know about it, how to write it. Please let me know. Ma'am, could you please change the medium of class? Yes, Tejaswini, we will do it on weekends. I think so. We will try to take it on MS Teams, but I am not completely sure. But I will try to. We have Shaurya Deep, ma'am, is the certificate of internship is yes, yes, yes. No, you will get the certificate for internship, but yes, you have to write the review paper and we have to send it up to publication. Okay, so we have to send it for publication. Yeah, Poshali, inborn errors of amino acid metabolism is quite good topic. Please, you can say proceed it and let me know.
मैंने कहा सर ये पिछले बताओ कैसे किया कहते हैं इसकी कितनी इंक्वायरी हो रही है तो फेल कर दूँगा मैंने कहा सर मुझे बताओ तो सही मेरे को पता था मुझे रिजल्ट मेरे पेपर में फेल हो तो रिजल्ट कैसे क्लियर कर दिया कहते हैं इतनी इंक्वायरी तो फेल कर देगा फिर कहते हैं मेरा रिजल्ट सर एक का हो गया दो का रह गया कहते हैं हम एक एक करके निकालेंगे
कौन है हाँ चलो मैं फोन पे बात करती हूँ ओके so yes no you can select any topic of your own point of interest and Rishta is saying while applying the course the time was mentioned 4:15. Yeah, dear, I understand, Vivita. From tomorrow onwards, we will take it on 4 p.m. by 4 or 4:15 p.m. I have my issues. I already told you the very first day. I hope you will be there. But it's okay. Tomorrow we will take at 4 p.m. Don't worry. I know you all have your classes, your practical works, and everything is there. So we will take it on 4 p.m. tomorrow. Okay. So Sambit is saying what is the last date? Then I today I have not mentioned you anything about the last date of submission of the review paper. Rather I told you 10th of December is the last date for submission of your topic along with the abstract. Getting my point? So So we will try to do it. We will try to compile the review paper. But 10th of December is the date only for submission of the abstract along with the title. Getting my point? So any of you have you selected any topic? Please let me know. Have you selected any topic? Are you doing it alone or you are doing it in group? Please let me know. So any of you are present here right now, please let me know. Have you selected any topic or not? Please let me know about the title because sometimes it's difficult for me, to just reply to your mails or even to the messages. Please let me know. Have you selected any topic or are you doing it alone or you are doing it in group? Please let me know. Ayushi, what's your topic is oral delivery of anti-cancer drugs. Okay. Please make the abstract and send it to me as soon as possible. Ayushi Krishna Pal, please let me know your topic. Please let me know your topic, please. Yes, Tejasmi, you have to select the abstract to me by 10th of December. Kalpesh is saying, ma'am, I selected the topic as Graves' hyperthyroidism in pregnancy. Okay, very good, Kalpesh. Please proceed it and let me know as soon as possible. Hari Amrit Raj, have you attended the previous lectures or not? My topic is diabetes. So Sambit, I will suggest you, please just not put it only a single word topic. Instead, you have to write the topic of 3 to 4 words at least. Getting my point? So you have to like the impact of diabetes in children, the impact of diabetes during COVID-19, something is like that. You have to make the title which is approximately of 3 to 4 words at least. So please let me know. Take your time, no issue, Sambit. Take your time and then let me know. Ayushi Krishna Pal Verma. Yes. So how mRNA affected by ketogenic diet and are also are useful in cancer treatment? Okay, very good Ayushi. Anamika, monoclonal antibody. Yes, it's okay Anamika. But try to make the title at least of 3 to 4 words, please. Yes, Pooja, I have your topic, I know. Radha is here. My topic is brain implants for drug delivery. Yes, very good, Radha. So, Radha, do you have classes today also? Do you have some practicals or something? Please let me know. Effect of eutrophication on benthic animals. Okay. Benthic animals or uh, benthic organisms? Okay, just send me the abstract and I will check it in briefly. Okay. Yes, you can take the topic and then all of you, it's my request to all of you that kindly share me your topics. Even share me the abstract as soon as possible. So now I have to tell you how to write the review paper. But I already told you this thing in the yesterday session also. Like you have to go on Google Scholar, all of you, or even on PubMed also. So on Google Scholar you can find the topic of your own point of interest. Like some of you are writing on force. We will take the topic of we will take the topic of this Anna. We are just taking their topic just for so that I will able to answer you. So we have to write review. So this is chemical meditation on marine organisms. So when you will open it, you will able to know like this is a paper by VJ Paul in 2004. So you have to open the PDF of this paper. Even you can do as much as it's possible, like 1 2 3 4 5. Even you can open page number 10, like chemical alarm signaling in aquatic predatory prey systems. You can take from 2016 also, prostaglandins in marine organisms, a review. So you have to open the review paper like chemical meditation of interaction among marine organisms. This was published in 2004. So you can compile their work together. Okay. So like this is, we have introduction, we have microalgilla, we have sponges. So this is the introduction. So you have to write the abstract. I don't know where the abstract of this paper is. So you have to write the abstract and then you have to continue it with introduction, and introduction there you have to briefly tell each and everything which is related to your own topic. Like if we will open this topic, prostaglandins in marine organisms. Like this is prostaglandins in marine organisms. This is also a review paper. So you have to write the abstract and you have to write the keywords which are there in the abstract. So the abstract is a nutshell of the whole paper. So this is introduction. Then we have the structure, biosynthesis and activity of prostaglandins in mammals. We have all the information. We have even have tables. So when you are adding the tables, you have to add the reference from where you have taken the data. Getting my point? So by this way you have to write the review paper of 4 to 5 pages at least. I hope little bit is clear to all of you. I hope it will be helpful to all of you to how to write a review paper. Please let me know. Is it clear to all of you how to select the topic? You can select any topic of your own point of interest. You can select it on COVID. You can select it on any topic which I provide you in the biochemistry lecture series. So you can select any topic of your own point of interest. So please let me know regarding it. Is it clear to all of you or not? Please let me know. Sorry, this is biochemistry. So please let me know. So you have to write at least 4 to 5 pages. 4 to 5 pages are mandatory for writing a review paper. Okay. So 4 to 5 pages are compulsory. No, you have to put the references just at the end of your paper. That means after writing everything, then you have to put the references, or you can say citations. You have to submit the title and abstract by 10th of December. Then I will give you more time for writing a review paper. I know all of you are new to this field, so I will not sorry. I know you all are new to this field, so I don't want to make it hodgepodge and everything like that. I just try to keep it very simple for all of you. Format, Diksha, the format depends upon the journal in which we will send the paper. Sometimes what happens, we just, like in my previous batches also, we just make the paper according to our own self. Format is there and review paper, references, but everything is dependent upon the journal in which we are sending our paper, because they have their own guidelines, they have their own patterns. So just keep it simple right now. When we will select our journal, then we will take their guidelines and then we will modify our paper according to it. Yes, Pooja, you can write it. It just depends upon you which type of review paper you are writing. Either you are writing it from the case study point of view. It just depends upon you. So I think that is all for the today's conversation regarding the review papers. I hope all of you have joined right now. Now it's 3:35. So now we will focus on our lecture which we have today. So tomorrow I will take the class by 4 p.m. or by 15 p.m. But I want to thank all of you to be there on time and to be there, and you all are interacting with me. So I want to thank all of you for this patience and everything. Yeah, Anjali, your topic is very good. You can take global climate change and its implications on coral reef ecosystem, and at the end you have to write a review. So yes, this paper will be definitely will be very good. So I thank all of you for your patience and for everything that you are here. Even we have some issues in the timing also, but till you are here, so I want to thank all of you. Now I clear all the doubts regarding the review also. You have to send me the title and the abstract by 10th of December, number 1. And then if you need some type of help, or you have some issues or even some doubts, you can mail me. My email ID is, you can mail me at any time, and even you can send me your abstract and title on this email ID. Cancers are related. Cancer is a very broad field, dear Diksha. Cancer is a broad field. You have to specify it on which cancer you want to work, whether you want to study breast cancer, you want to do it on ovarian cancer. It depends on you, basically what you want to convey to all of us. Please let me know. Kalpesh, you have to put the whole references just after completing your full review paper. And yes, when you are taking the line from some another paper, then at the end of that particular line, you have to add the reference number only. Let me show you. Like here, let me show you. So if you are able to see here, they have taken the line, this one, fluorotannin concentrations increase in alga exposed to UV radiation. So at the end they are mentioning 50,112. Are you able to see it? Let me make it more elaborate. So at the end they just, after the line, they just add the reference with number only, 50,112. That means the line has been taken from the reference number 50. Let me show you the reference number 50 now. So at the end we have references. So this is the reference number 50. So we have so much of references. So this is the reference number 50, H. pavia. So from this paper, this particular review paper, or you can say this particular paper, they have taken this line which I just showed you right now. Similarly here also. So you have to add the number only at the end of the line, and the references will be right after the conclusion. So these are all are the references. Getting my point? Is it clear to you or not? Please let me know. Is everything is clear to all of you? Please let me know. So you have to put the references just at the end of your paper, but you have to add the number, like number 1 reference, 2, 3. So you have to add the numbers. So I hope this will help you. Even if you have any doubt, you can let me know at any time. You can message me, and even you can mail me also. So this is my mail ID. Please let me know if you have any problem. Okay, so good, it's now clear to all of you. Now we have to move further for our today's day 2, which we have to do structure of DNA, RNA and central dogma. I know you all must know about all these things. Yes, I give you one homework also. I asked all of you to make the structures of purines, pyrimidines, nucleotides and nucleosides, and you have to click those pictures and upload it on Telegram. But any one of you, I don't think so, you have put the images on Telegram. Please let me know if you draw the diagrams. I don't think so. Yes, you didn't paste any diagram or something. I just told you yesterday that you have to draw the diagrams of purines, pyrimidines, nucleotides and nucleosides, and you have to upload all those diagrams on Telegram. But I think you all are not serious what I said already. So please give me reply why you are not upload it on Telegram. Is it difficult? Please let me know. Or you are just busy on selecting the review paper. I think you all are busy for selecting the review paper. If any one of you have drawn it, please share it with me either on my Telegram group or even on WhatsApp. We don't have a WhatsApp group, right? Okay. So you have to upload it, please. If you draw the diagrams, please upload it on Telegram. I will definitely check your diagrams. Okay. Okay. So it's my request to all of you, please upload the diagrams, and we will try to make the WhatsApp group also, because WhatsApp group is little bit convenient. So that is why. Yes, I know all of you are busy in selecting the review paper, not review paper, all of you are busy in selecting the title basically. So no issues. So today we have to continue with our previous lecture series, which is on nucleosides, nucleotides. So today we have to do the structure of DNA, RNA and central dogma. So we have to continue with the presentation which we have yesterday also. Yeah, from here. So this is the structure of the DNA and RNA. We already discussed it yesterday also, that the DNA is double stranded structure, RNA is single stranded structure. The nitrogenous base pairs, they bind with the another nitrogenous base pairs with the help of hydrogen bonds. So we have cytosine, guanine, adenine, thymine in case of DNA, and in case of RNA, instead of thymine we have uracil. So cytosine, guanine, adenine and uracil. Instead of thymine we have uracil. We already discussed it yesterday. So this is the DNA, which is basically deoxyribonucleic acid. It is basically the segment of particularly the segment of a DNA, or you can say portion of DNA are called genes. So in the nucleus is present where? The nucleus is present inside the cell. So inside the cell we have a nucleus. Nucleus consists of chromosomes, and on the chromosomes we have the genes, and genes are basically the segments of a DNA. So the DNA is there present in the form of genes with just yes. So inside the cell we have nucleus which consists of chromosomes, and on the chromosomes genes are present, and genes consist of particular segment of DNA. So DNA is tightly wound into chromosomes and located in the nucleus of the cell. DNA cannot leave the nucleus, so DNA must be present inside the nucleus. So DNA is double stranded. DNA is double stranded, you all know it, and RNA is single stranded. This is RNA. So RNA is single stranded and does not have to stay in the nucleus. So the DNA always remains inside the nucleus, but RNA is single stranded and is able to just move out of the nucleus and to be there in the cytoplasm. So RNA is not found in the chromosome because it does not carry the genetic code. However, it can read the DNA code and take the information out of the nucleus. So RNA's main job is to build the proteins only. So that is why RNA moves out of the nucleus and it takes the information out of the nucleus. So all the information is present inside the nucleus of the cell in the form of a DNA. From DNA we will get the RNA, and the RNA then will move out from the nucleus into the cytoplasm, and RNA's main job is to build the proteins. So this is the DNA structure. We already discussed it yesterday also. The building blocks of DNA are called nucleotides. One nucleotide is made up of three important things. We have 5-carbon sugar, which is deoxyribose. Phosphate group, we have nitrogenous bases. So we have the building blocks of DNA are called nucleotides. Then we have RNA structure. The building blocks of RNA are called nucleotides just like DNA. But in case of RNA, instead of deoxyribose sugar, we have ribose sugar. Getting my point? Here we have 5-carbon deoxyribose sugar, and here we have ribose sugar instead of deoxyribose. Along with it we have phosphate group and we have nitrogenous bases. And one more thing, instead of thiamine we have the uracil. So DNA is double stranded, like here they have the hydrogen bonding between the nitrogenous base pairs, and RNA is single stranded. DNA always remains inside the nucleus. RNA moves out and just pass the information from the nucleus to the whole cell of the body. So both DNA and RNA are single stranded, contains the same 4 nitrogenous bases, have the same 5 carbon sugars, contain the phosphate groups also. So this is DNA and RNA. Yes, now we have another thing, which is the DNA structure. So DNA structure can be discussed as primary structure, secondary structure, tertiary or quaternary structure. So the order of bases on the polynucleotide sequence, the order of bases specifies the genetic code. So primary structure is a very basic structure. Let me show you. So this double helical structure is the very basic primary structure. After it we have the secondary structure, which basically consists of three-dimensional conformation of the polynucleotide backbone, which is equal to the double helix structure. Then we have a tertiary structure. In tertiary structure we have the supercoiling of the, you can say, DNA polynucleotide chain, and in the quaternary structure we have the interaction between DNA as well as proteins. So this is the secondary structure. Now we have the tertiary structure, which is highly in which there is highly the coiling of the DNA chain basically. So it is circular duplex DNA. So this is relaxed and this is supercoiled DNA structure. So each cell contains about 2 meters of DNA. DNA is packed by coiling around a core of protein known as histones. Yes. So in case of tertiary structure of DNA, what we have basically, we have the supercoiled DNA. This one, we have the supercoiled DNA, and along with it we have the core of proteins known as histones. So we have proteins also present which are known as histones. So the DNA histone assembly is known as nucleosome. When there is an interaction between the supercoiled DNA and the histone proteins, it is known as nucleosome. So histones are rich in lysine and arginine residues. So histones are basically rich in lysine and arginine residues. Now we have the quaternary structure, four standard form of DNA, role in regulating and stabilizing the telomeres and in regulation of gene expression. Small molecules such as porphyrins, they are present to stabilize the structure. So the quaternary structure is quite complex. And now we have the types of DNA. We have A DNA, B DNA and Z DNA. We have little bit difference in all these types of DNA. Now again we have DNA structure and function. But let me know, is it clear to all of you? Everything is fine till now? So today we have discussed briefly about the DNA, the types of DNA, the structures which are there. We have primary structure, we have quaternary structure, tertiary structure. So you have to know about the primary structure and the tertiary structure of DNA is quite important. So this is the structure and this is the DNA structure, or you can say the functions of DNA. DNA structure is well known double helix form by Watson-Crick's base pairing of C with G and adenine with thymine. This form is known as B form of DNA and it is the most favorable and common state of DNA. It is highly specific and stable. Base pairing is the basis of reliable genetic information storage. So this is the famous Watson and Crick model which describes the structure of DNA, that the DNA is double helical structure. We have minor group, we have major group. I will not discuss this structure. This is a very common structure of DNA. All of you must know about it. But yes, this tertiary structure is quite important. In this tertiary structure, when the DNA is supercoiled, getting my point? Here the DNA is supercoiled. DNA is packed by coiling around a core of proteins known as histones. So we have the proteins which are present and whole this DNA histone interaction is known as nucleosome. So this histone DNA interaction is known as nucleosome. Histones are rich in lysine and arginine residues. So this is a Watson-Crick model again. This is again the base pairing that adenine will bind with thymine, cytosine will bind with guanine. The hydrogen bonding is there. We already discussed it before also, that there is a hydrogen bonding. This is again the Watson-Crick base pairing of C with G and adenine with thymine. Now we have another term which is replication of DNA. Now what is basically the replication? Replication is the copying of the DNA. So from one DNA we are able to get the copy of it. So it is a process by which DNA is copied itself by own. So this is the replication. Two polynucleotide strands running in opposite directions and coiled around each other in a double helix. The strands are held together by complementary hydrogen bonding between the specific pairs of bases. So we have the complementary hydrogen bonding. So this is replication, transcription and translation. This whole process from DNA to RNA and from RNA to proteins, this whole process is known as central dogma. Let me show you where there is the central dogma. Now they are just discussing about the structure of RNA, the functions of RNA, the types of RNA, like we have mRNA, tRNA. I will share this PPT. I hope you will read this by your own self also. So this is the central dogma on which I am focusing right now. From DNA we are able to get mRNA, and from mRNA we are able to get protein. This whole process is known as central dogma. Okay, so transcription and translation is included in the central dogma. Is it clear to you, or do you have any query? So, we will try to discuss transcription and translation in detail. Have you done this before also? Please let me know. Otherwise, we will have to discuss it very thoroughly. Have you done transcription and translation before also? Please let me know. So have you done transcription or translation before? Any one of you, please let me know what is transcription and what is translation. What is the difference between transcription and translation? Any one of you, please let me know. What is the difference between transcription and translation? Any one of you, please let me know. Okay. So Kushali, please let me know the difference between transcription and translation. Okay. Any one of you, please let me know about the difference between transcription and translation. What happens in transcription basically? Yes, very good. Transcription is the process of getting mRNA from the DNA. And translation is from messenger RNA to proteins. Now you must know that some factors are also involved in the process of transcription and translation. Some factors are also involved. Yes. So you all are aware of this central dogma. And what is the reverse of central dogma? What is basically reverse transcription? What is reverse transcription? Any one of you, please let me know. What is reverse transcription? What is, yes. So what is reverse transcription? Yes, very good, Ayushi. So you all are aware of all these terms. Okay. So this is the very basic things which we have done in the 2 days. We have done DNA, the structure of DNA, the types of DNA, the difference between DNA and RNA, what is central dogma, which is basically from DNA to we get mRNA, and from mRNA we are able to get the proteins. So I will share this presentation of 177 slides. I will share it with all of you. You have to read it once. You have to go through with all these, you can say, slides, and let me know if you will not able to get anything. So this is all about the mRNA. Then we have the proteins which are there, which are associated. So proteins are made up of a chain of amino acids. Proteins are enzymes which catalyze and regulate the chemical reaction. So these all are proteins. The steps are there which are involved in making the protein, which is basically translation. So in the protein we are just dealing with the translation only. So I will share this presentation with all of you. I hope all of you will be able to get it. If there will be any problem, then please let me know, then we will discuss it again. Getting my point? If you will get any problem regarding it, then please let me know. We will discuss these process again also. Even I have one video of central dogma and this transcription and translation also. I will share those videos with you. You have to go through it and let me know. If you are not able to get anything, then please let me know. So is it clear to all of you? Please let me know. Is it clear to all of you? Please let me know. Okay, so tomorrow when we will meet, I will check in the group also the structures of purines, pyrimidines, and about nucleotides, nucleosides. And tomorrow, yes, Sandra, the application is there only for in case of DNA. Why? Because DNA is double stranded. From double stranded, we are able to get the replica of that particular parent sequence. But in case of RNA, it is not possible. So that is why we just focus on replication in case of DNA. So replication, transcription and translation, they are very important in their own perspectives. So tomorrow we will discuss the main thing which we have to start. We have to start metabolic and hormonal disturbances. This is very important topic, metabolic and hormonal disturbances. Which is the main cause of stress these days, which is the main cause. That's why we are sometimes very happy and why we are sometimes very much depressed, we are very much angry, or we are very much sad because of all these hormonal issues. Getting my point? So we have to study in detail all these hormonal imbalances and how these hormones, they play a very important role in our life. So I always told sorry. So I always tell my students to be happy always. You should be happy from inside. If you have any type of stress in your life, then your mental health, your internal health will not be as good as it should be. So I also suggest all of you to also, to be remain calm and to be remain happy. Don't take stress of anything. This is life. Something good is happening, something bad is happening, but don't take the stress of anything which is there. Try to remain calm, or you can say try to remain happy always. So I hope this is all for today's lecture series. Tomorrow we will start the hormonal imbalances. Till then you have to let me know the topics and the problems, the abstracts, or if you have any query, you have to let me know it. Getting my point? Is it clear? please let me know. And tomorrow we will meet. Yes, I will send the PPT on the Telegram. No issues at all. I will send it. And tomorrow we will meet on 4 or 4:15 p.m. Is it okay to all of you? And on Saturdays and Sundays we will take 1 hour extra. Getting my point? On Saturdays and Sundays we will take 1 hour extra. So I hope it is clear to all of you. So we will meet tomorrow, and on weekends, that is on Saturday and Sunday, we will take the 2 hour long session. Everything is fine? Is it okay to all of you? Please let me know. So that's all for today's session, and we will meet tomorrow at 4 p.m. Any other thing you want to know, please let me know. If you have any query, any question, please let me know, all of you. If you have any query, any question, okay. So please let me know if you have any query or something. Otherwise, we will start hormonal imbalances tomorrow. And be ready for the extra classes on weekends. Okay, so we will try to do the extra classes on weekends. No, Narita, MBBT and biochemistry classes, they are totally different. And right now we are taking the biochemistry classes. MBBT class is at their own timing, which is 9:15 p.m. Actually, basically, I put the wrong, you can say, uh, by mistake I put MBBT here, but it is biochemistry session. But if you join with no issues at all, we will meet at 9:15 p.m. today. Yes, Sandra, please proceed with the topic. You can proceed it. Just send me the abstract as soon as possible. You have to send me the abstract as soon as possible. So you have this weekend along with you. You have Saturdays and Sundays. Try to select the topic and try to make the abstract on these weekends. Please, all of you. Oh, you missed yesterday's class on MBBT also. But be there on time today. We will try to take it on. We will try to take it on 9 p.m. or 9:15 p.m. Be there in the group, please, if possible. But no issues at all. We have not discussed much. Today we have to discuss in detail. Okay, so no issues at all. No problem. Don't worry. Sometimes it happens. No issues. So all of you, you have weekend along with you. You have 2 days, Saturdays and Sundays, and not Sundays, Sunday only. So you have Saturday and Sunday, and you have to write the abstract, and you have to send it to me. Okay. Take your time, no issues at all. But just let me know what you all are doing. Okay. Have you done hormones before also? Any one of you, have you done hormones before also? Like the, you can say, abnormalities which are there because of hormonal dysfunction or something like that. Have you done it before also? Any one of you, please let me know. Okay. Okay, Ayushi has done little bit. Okay. So tomorrow we will discuss it in detail and I hope all of you will get it properly. Okay. So we will meet tomorrow. Till then, take care of yourself. Okay. So bye bye from my side. We will meet tomorrow. Yes, Pooja has sent me their chat. Pooja, I will try to read it today by night. So I will try to reply you as soon as possible. Okay. So that's all from my side. Take care of yourself, all of you. Bye bye. Okay, so take care of all of you. We will meet tomorrow.